Hi, doctor friends. Welcome to my YouTube channel PG Doctors Indranju. Today's topic is about the morphology and physiology of the bacteria. The first of all, let's start with this topic. It is very important for the bacteria which are prokaryotes, eukaryotes. It is very important to know about the bacteria. So let us start with the prokaryotes. What is the prokaryotes? Prokaryotes, it is the first living organism which evolved to becomes a eukaryote organism. First, when the evolution being starts, then first organism is becomes the prokaryotes, and it is first of all the characteristics and what is the main feature of the prokaryotes, so that you understand and you can differentiate the prokaryotes and eukaryotes. The prokaryotic characteristics is it has no any organelles found. That means organelles, that means no nucleus, no nucleolus, no mitochondria, no golgi apparatus, and endoplasmic reticulum, lysosome, this all are absent. Due to the all are absent, so the mitotic division can't be possible. And here the deoxyribonucleotides, pinocytosis, cytoplasmic streaming, and withdrawal in the membrane are absent. This is the basic feature of the prokaryotes. That means, and it divide by binary fission, not the mitotic division, and it is always stay in the single, not in the multiple and the polycell or multiple cell. Here all things are organelles. Here the all things are absent, all organelles nucleus. So it depends on other organism or other cell for their life process. Due to here the mitochondria are absent, so the energy requirement of the such prokaryotes depends on the other cell. Golgi apparatus, endoplasmic reticulum, all are absent. So it requires many cellular function, and its requirement is fulfilled by the other cell. So it depends on other cell always. And the genetic material of these both are DNA and RNA both, with chromosome is single and circular. Example, bacteria, gram positive and gram negative bacteria are the prokaryotic structure, including chlamydia, mycoplasma, and blue green algae. This is the prokaryotic structure. Next one is the eukaryotes. What is the eukaryotes? Eukaryotes, first it evolved from prokaryotes, and all organelles which are absent in the prokaryotes are present in the eukaryotes, and it divided by mitosis or meiosis, and it may be stay present in the single or multicellular structure. And the genetic material, DNA, RNA both, and chromosome, and is more than one and linear. Here the example, algae, fungus, protozoa, all are the example of the eukaryotes. Here the one thing you have to remember, that the bacterial genetic material is both DNA and RNA, but in the virus only DNA or only RNA single is present, either DNA or RNA, not both. Next topic is about the important structure in the bacterial cell. Important structure start with the cell envelope. Cell envelope includes the cell wall. All things are mentioned in this topic. Cell envelope. What is the cell envelope? Then you starts with the outer membrane, then cell wall. It is from outer to inner. So outer membrane is outermost part, and then after cell wall, then after periplasm, and then after the cell membrane. If I draw such type of the diagram, it is the cell. Then around that, this membrane is the cell membrane. After that, this is attached membrane is periplasm. After that, a thick a thick wall is cell wall. After that, a another membrane is this is the outer membrane. This feature this feature is present in gram bacteria. Where outer membrane is present or absent, I am going to detail about that. So first of all, you have to remember it is outermost and it is the innermost layer of the cell. First start with the outer membrane. Outer membrane, I am showing the diagrammatic representation of the gram positive and gram negative so that you can easily differentiate. So the outer membrane is present only in the gram negative bacteria. So gram negative, you have to remember that the outer membrane is present only in the gram negative bacteria, and the outer membrane, which are composed of the lipopolysaccharide. That means the glycerophospholipid. It is the asked in the MSPG that what is the lipopolysaccharide present in the outer membrane of the gram negative bacteria. That is the glycerophospholipid. Okay, you have to remember that lipopolysaccharide is glycerophospholipid, and it is the bilayered membrane, and protein in that structure is known as the porins. So I'm showing all thing in that diagram so that you can easily understand. Here the outer membrane, after that it is the cell wall. That means cell wall is made up of the peptidoglycan, which has the cross peptidoglycan, also known as the murine, and it is made up of the cross-linking structure of the N-acetyl glucosamine. You have to remember N-acetyl glucosamine and N-acetyl muramic acid. So these both structure are cross-linked to make cell wall structure. So it is present in the both gram positive as well as gram negative bacteria, but it is the cell wall which are absent in the mycoplasma. Okay, so this structure are absent in that cell wall. First, without wasting of the time, the diagrammatic representation of the envelope is here. If you look this image, I have already shown that the water structure is present outer to inner. Here the if we are look about the gram positive bacteria, this diagram showing that it is the it is the structure inside the cell, and you can easily make a like cell structure, and it is the membrane is the plasma membrane. This membrane is the plasma membrane. Okay, then after the this space I'm changing color so that you can easily understand. This structure, this space is called as the periplasmic space. Okay, and outer membrane, it is the outer membrane which are known as the peptidoglycan, made up of the cell wall is made of the peptidoglycan, and here the it is the layer of the peptidoglycan which are multipolymer of cross-linking structure of the N-acetyl glucosamine and N-acetyl muronic acid. The here the you can easily see that it is the I am make this star like structure so that you can differentiate from gram positive and gram negative bacteria. Here the star like structure is called as the teichoic acid, and you first of all I'm describe my things about the gram positive gram negative, then I will differentiate such the here this is also make a diagrammatic representation, that means it is the plasma membrane. Then after here the periplasmic space, then after periplasmic space, then cell wall. This is the cell wall, this is the cell wall, and after that another structure is the periplasmic space. After that this structure is the lipopolysaccharide, which are the outermost part or outer membrane. It is here the channel is found between the outer environment and in the outer periplasmic space, which channel make it permeable to the solute electrolytes in the lipopolysaccharide membrane. This is known as the porins for transport of the electrolytes, solutes, etc. Here first of all, how can you differentiate? That is the gram positive bacteria and gram negative bacteria. Gram positive bacteria has only 3 layers, that means the plasma membrane, periplasmic space, and the cell wall, that means the peptidoglycan, which contains teichoic acid. And here the cell wall is thicker, that means it is of 20 to 80 nanometer in the thickness, while that in case of the gram negative bacteria, this thickness is very shorter, very small compared to the gram positive bacteria. So you have to remember gram positive is the 3-layered structure, and outer membrane is thicker, which contains teichoic acid. While that in the gram negative bacteria, here the periplasmic space is outer and inner. In between that, the cell wall is a thin layer of the cell wall is consist here, and the outer membrane is the lipopolysaccharide. Okay, you have to remember such difference. Next is about the cell membrane. Cell membrane are the mainly semi-permeable membrane, known for the semi-permeable membrane structure. And the also known, also you can say it is the selective permeability. So it permeate the electrolytes, selective electrolytes. Next, the inner cell membrane is the infolding structure, which are also known as the mesosome or chondroid. You can also call it as the chondroid. It is the chondroid. Chondroid is actually, it is the uh, principal site of the respiratory enzyme, due to consist of respiratory enzyme in the bacteria. So it is analogous to mitochondria in the eukaryotes. It is analogous structure to the mitochondria, and it formed by the, if you make a diagram, the cell, it is the outer membrane. This is the, if you look, the, it is the plasma membrane. Then here the structure is enfolding, which are known as the mesosome or chondroid body, which are responsible for the ATP generation or ATP generation or energy generator, you can say it. And one thing is that the cell membrane of the plasma membrane of the mycoplasma is the estrol. That means only bacteria containing estrol in the plasma membrane are the mycoplasma. Okay, this is membrane present, estrol containing membrane present in the only eukaryotes, not in the prokaryotes, and only mycoplasma is that prokaryotic structure which contains esterol. Next thing is that the capsule. What is the capsule? It is the protective structure of the prokaryotic organisms, which are found in both gram positive as well as gram negative bacteria, in not in all cases, in some. It is around the cell envelope, which is secreted by bacteria itself, and it is viscid substances, and also a slime layer made up of the simple sugar or polysaccharide. But in the Bacillus anthracis, it is the polypeptide D-glutamic acid, and E. coli, it is the peptidoglycan and muronic acid. The capsule is, it is the antigenic in the nature, but protection from the phagocytosis, it is due to protective nature. So it in the bacteria, due to presence of the capsule protects from the phagocytosis, from the lytic enzyme, and for the desiccation. If you look this diagram, the some difference between the capsule, slime layer, and the biofilm. So the capsule is like a regular structure and a regular pattern, and slime layer is the like irregular structure, and bacteria in that means bacteria secrete a slime layer, capsule and thin biofilm. These are different in that the capsule and slim layer, the bacteria secrete, single bacteria secrete its capsule, and while that in the biofilm the many bacteria secrete biofilm and becomes a make a protective structure. And in the capsule and the slim layer, due to it is made up of the simple polysaccharide, so it is also known as the glycocalyx. Okay, first of all, I'm showing you the image of uh, this so that you can easily differentiate. This is the structure of the bacteria. If you look at this side, this structure here, the cytoplasm is there, and some red type of the granules which as the ribosomes, and here the nucleoids or DNA are present. Around that is the plasma membrane, which is it is the 3D structure, so that you can show the what is the exact structure of the prokaryote or bacteria. And here it is plasma membrane. After that the cell wall, around that a protective structure are formed known as the capsule. Okay, the lab method to differentiate the capsular structure, then the India ink, the lab method that differentiate India ink, the clear halo of the capsule showing you in the microscope. It is due to the India ink can't penetrate the capsule, so in capsule becomes the clear halos. Second method is the Mendel's capsule stain. This also can't penetration to the capsule, so it is also clear halo structure. Another method to detect the capsular structure is the serological method. In the serological method, here the capsular swelling occurs known as the Squillens reaction. It is due to the capsule, already told that the capsule is the antigenic in the nature, so selective antibody react with them, and it is becomes the permeable to the water, so that it becomes swelled, and also known as the Quillen's reaction. Especially it is found in the pneumococcus. The Gram stain. The Gram stain is not for the capsule, due to it is not visible due to very little affinity with the basic dye to the capsule. So next is the where in which gram positive or gram negative organism which are capsulated. This is important for your second year and for PG exam. The bacteria which are capsulated, first of all start with the gram positive bacteria, gram negative bacteria. Gram positive bacteria, the Bacillus, Megatherium, Anthracis, these are capsulated organisms. Streptococcus pyogen due to hyaluronic acid capsule, and Streptococcus pneumoniae, Streptococcus agalacti, Staphylococcus epidermidis, Staphylococcus aureus, Clostridium. These are the gram positive organism. Okay, next is the gram negative organism. Here the gram negative organism is For the capsule, gram negative organism most common. That means capsule most commonly found in the gram negative bacteria. That means E. coli, Neisseria, Klebsiella, H. influenza, Pseudomonas, Salmonella all contains capsule. That means capsulated bacteria. Another is the fungus Cryptococcus neoformans. It is the yeast which are also capsulated, but it is similar to capsule. Okay, this is asked in the All India 13 PGI and also uh, also in the EMS. So thanks for watching my video. If you want to advise anything about my video, to reform what is wrong, what is right, and any interest, please write in the my comments box and watch my video till last so that you can understand all things. Thanks for watching my video. Have a nice day.